Ani Novianti. Please, Miss Ani.
Okay, thank you, Miss. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. And today we do a seminar, and the material is genetic engineering in plants tissue and the engineering in plants by Ilfa Qurrota Aini. Miss Ilfa, are you ready?
Yes, Miss Ani.
Okay, thank you. And this time is yours.
All right, thank you very much for the time. So hello everyone. My name is Ilfa Qurrota Aini, and for this seminar, I will talking about genetic engineering in plants. Wait a minute, I will share my screen first. Can you see the screen?
Yes.
Yes.
All right, so let's start. Let's start the talking about genetic engineering in plants. So for the first, there's a definition of genetic engineering in plants. It means that there is a copies of genes from the organism with a desired trait, or the edge of genes to a single plant in cell laboratory. So we can say that there is a genetically modification in the DNA or gene of the plants. So there will be a new characteristic, or maybe it will be more resistant of something. It will be like that. So for the steps of the process of genetic engineering in plants, the first here there is DNA extraction. This process accomplished by taking a sample of bacteria containing of the gene of the gene interest in, and taking it through the series of steps that spare the DNA for the other parts of the cell. So because we only need the DNA extraction, we have to we only need the DNA, so we have to extract the DNA to do the genetic engineer to do the modification. For the second steps, it's gene cloning. It's about located and copied the gene. So because in this step there will be we will be separating the single of gene of the interest from the rest of the gene extracted. And in this step also, because in the next stage there will the scientists will need some will need the gene for the further studies. So in this stage of the gene cloning, they will be make like thousands of the copies of the DNA. So for the third step, it will be gene modification. In this process, the gene will be modified to express in a desired way by altering and replacing the gene's region, and it will be done in the test tube by cutting the gene apart with the enzymes and replacing certain regions. For the next step, it's transformation. In these steps, the genes will be delivered into the tissue culture cells. It can, in this step, you can use several methods, such as several methods that will be include some other organism like agrobacterium, microfibers, and other method like agrobacterium, microfibers, and electroporations. And for the main goal of this is to transport the new genes and deliver them into the new nucleus of the cell without killing the gene. For the last step, it's back crossbreeding. The transgenic lines are crossed with elite line to make high-yielding transgenic lines, and it's the final parts of producing a genetically engineering crop. So, but this genetic engineering in plants have some limitations. So, not every plants can be modified. The DNA can be modified. The first limitation is the breeding only can be done between the two plants that sexually mate, and the second one is when the plants are mated or crossed, many traits are transferred along the trait of the interest, including the traits with undesirable effects on the yield potential. The example of the example product of the genetic engineering in plants here are the first. There is poplar trees, which can fight the pollution. It's made by the scientists from University of Washington. The poplar trees will absorb the polluted water through the roots and clean before the water is really released back into the air, and it will be, and these plants were more efficient at cleaning than the regular poplars. The second example is golden rice. It's the goals of making the golden rice is to make the healthier food, and also the vitamin of the golden rice is the same with the carrot. So when we eat the golden rice, you will get the vitamin like you are eating carrots also. And the next one is non-carrying ions. It's made in New Zealand at 2008, but it's like the non-carrying ions means when you cut it, you won't cry, but because the original ions, when you cut it too much, you will feel like crying because of the spicy from the ions. And the last example that I will say is pesticide resistant repeat plants. It's plants that used to make certain types of vegetable oil. Besides the many example of the example of the product of genetic engineering in plants, but there are also pro and contra of doing the genetic engineering in plants. So for the the first pro is the more nutrition in plants. So like because there is the genetic modification in the plants, there will be like more characteristic or more nutrition in one plants, and also the plants will be more resistant to the disease. So it won't be like easy to get the disease or easy to die because of the disease. And the last pro is produced to improve the seeds. For the contra, because many of people say that the consume the genetic engineering products, plants will be danger because there will be maybe some pathogens that adapt to the new genetic profiles, and also there will be unexpected negative side effects like healthy effects or any others that can't be controlled by the scientist. So I think that's all.
Okay, thank you, Ilfa. I open 3 questions for this session. Any question for audience? Okay, this is we have Felicia and Miss Lilith for the question, and the first is Felicia.
Is my voice clear?
I'm sorry, Felicia, I cannot hear you. Okay, it's okay. And Felicia question in the chat box, and maybe in Miss Lilith question.
Okay. Thank you, Miss Ani. Thank you, Miss Ilfa, for the great presentations related with the genetic engineering in plants. I would like clarify about the steps in here. Number 3, this is the step is gene modifications. When we relate with the example, I see in here the number 1, the example number 1 is golden rice. May you explain more what is the modification in gene? What gene that can modify to produce golden rice, Ms. Ilfa? So this is the example of golden rice. And the steps is, one kind of the steps is gene modifications. My questions is, what is the gene part, or maybe what kind of the gene that can modify, and the product is golden rice. That's the different with the common rice, right? Thank you, Miss Ilfa.
Okay, thank you, Miss. And Valisa's question is, do you think that genetic engineering is easier in plants compared to animals? If it yes, what is particular reason? Miss Ilfa, have you the answer?
Wait a minute. I need more time.
Okay, from audience, have you question? Okay, dia. Dia, what is your question?
Aini, so I have a question about the effects of genetic engineering. So is there any example of harmful effect of genetic engineering in food? And maybe you can show the real example of the harmful effect of genetic engineering. Thank you.
Okay, thank you, Miss Dia.
I will try to answer Miss Lit question first. So for the golden rice, the modification is in the gene. It's add the genes that can make the rice plants produce the enzyme that seems like beta carotene in the vegetable. So in the the golden rice will have some vitamin like vitamin in carrots by adding the genes from the other plants to the plasmid. Is it clear, Miss?
Okay, so I can get the point from you. So for the gene modifications for producing golden rice, the scientists insert beta carotene. The beta
Yes.
carotene that comes from vegetables. So the golden rice that's different with the common rice. When we eat the golden rice, that means we can also eat the beta carotene.
Yes.
Okay, thank you, Miss Ilfa. Thank you, Miss Arni.
Right. So for Felicia's question, is it easier to do genetic engineering in plants compared to animals? The answer is yes, because plants have totipotency totipotency scale. We so wait. So it will be to it will be easier to see the success in plants because the thought. Right. I'm sorry. Wait. I will start again. So the genetic engineering is easier in plants because the plants have totipotency properties. So it make each part of the plant cell is can make the new individual. So the successful of the genetic engineering will be greater than the animals. Is it clear, Faisa? All right. Thank you. Maybe I need time for answering this question.
Is their question is the fact of genetic engineering, especially harmful genetic engineering in food, and show the example. So we waiting for answer. For our audience, have question again. Okay, Nabila. And we have one left question. Please, your question, Nabila.
Okay, thank you, Arni, and also thank you, Ilfa, for the presentation. So, what I would like to ask now is, as we know, some of the genetic engineering seems like unethical to the natural, right? Like, um, like, how is it? Editing on human embryos, is it? That seems like unethical to the natural. So for this case, is there any limitation of genetic engineering? So that's my question. Thank you.
Okay, thank you, Nabila.
All right, so I will try to answer Priya's question. Is there any harmful effects from genetic engineering? Actually, the scientist was explain about it before, because there is maybe something like toxic toxic chemical, toxic things, or maybe bacteria that inside of the foods that we eat, that genetic engineering food. But because 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 a scientist do do many trials and error before before say that this food is safe to eat. So until now, there is no case about the side effect of consuming the genetic engineering food. But maybe there is some uh, allergic reaction from people that uh, because of the, what should I say? Because in the original one, there is no things like that, but in the genetic engineering food, there is things like that. So because the person doesn't know there is the things that can make him or her allergies, so maybe there will be allergic reaction of consuming genetic engineering food. And for Bella's question, some of the genetic engineering seems like unethical to the natural, such as editing human embryos. Then there is any limitation in using genetic engineering. So for the limitation, maybe for there, it's not stated in something like the rules that scientists have to taati to make to do the genetic engineering. But as long as that the genetic engineering is the scientists do genetic engineering not for making something dangerous, if the embryo of the human can be modified, be, if there is something wrong in the human, like maybe like there is a disease in the blood cell, or some, or yeah, that's the genetic disease. Yeah, it's like that. Is it clear, everyone? I'm sorry. Oh, yeah, I got the point, Ilfa. Thank you.
Okay, one question again. Any question? Okay, Diki.
All right, thank you very much for the opportunity. So my question is that, as earlier said, Ilfa said that genetic engineering in plant gonna have a higher success percentage than animal genetic engineering. I mean, that's pretty much understandable because maybe plants much more easier to manipulate than the animals because plants that doesn't need a lot of taking care of the plant itself. But my question is that what is gonna be the main factor of failure in genetic engineering on plants? Is it like human failure, or there is like criteria to make the plants gonna be successfully genetic engineered, something like that? That's my question.
Okay, thank you, Diki.
All right, for the next question, the main failure of of the main factors that make the failure of genetic engineering in plants is because the gene that will be inserted into the nucleus was dies, or turns out that the gene is not matched with the plants with the nucleus that will be modified. The gene will be modified.
All right, thank you very much. That's pretty much understandable.
Okay, thank you very much for the audience question and the for answer. And maybe I think it is enough for this session. And I have the conclusion. Genetic engineering copies a gene from an organism with a single plant cell in laboratory. And this is the same basically in genetic engineering, but the Ilfa topic in plant and has 4 steps. There are DNA extraction, gene cloning, gene modification, and transformation, big crossbreeding. And the one of the example is insecticide corn, golden rice, and etc. It is the genetic engineering in plants. And genetic engineering doesn't have harmful input because yeah, we can taste before we producing. And in plant more successful than animal because yeah, the process in genetic engineering in the process. Yeah, I think it's enough in this session. Thank you very much, Aikos. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.